Alright, you guys, it's Jubilee here, and I'm back for another TA review. This week is quiz 6, and we're going to be covering the lymphoid system. And hopefully by the end of this, you'll be able to tell me all about these pictures up here. So we've just got some examples. This is going to be the spleen, the thymus, and a lymph node. But yeah, hopefully by the end of this review, you'll be able to not only identify these organs when you're shown a picture of them, but tell me a little bit about them, some of the structures that are found within them as well. Okay, so functions of the immune system, right? So that's what we're covering this week, the immune system. So hopefully you kind of know the basics of the immune system from biology or a similar course, but we are going to go over them just a little bit. So the immune system, right, is designed to protect you against foreign invaders or antigens, right? So an antigen is basically anything that your body recognizes as foreign, as not a part of your body. So yeah, it's supposed to protect you against that. And then it also helps to produce and protect a germ-free environment, right? So if there are any antigens, any foreign invaders, any germs, anything like that, not only is it going to protect you against those germs, but it's also going to get rid of them, right? To produce that germ-free environment. And so there are many ways your immune system does it. It kind of does it in three main steps that we're going to break things down into. So the kind of first line of defense when it comes to combating infections is going to be, number one, breaking the cycle of transmission. Okay? And so that basically just means it'd be great if we can fight off infections or anything, but it'd be even better if those infections didn't happen in the first place, right? If all those germs, those bacteria, those viruses couldn't get into your body where they can cause harm in the first place, right? And so that is why we have all of these physical barriers, right? That's why you have your skin. So that's what this is depicting right here. All right, so that's why you have your skin. That's that tough barrier that hopefully prevents things like bacteria, like viruses, fungi, anything like that from getting into your body where it can cause harm. You've got things like your mucous membranes, right? So like the mucous membranes in your nose, in your mouth, your eyes, everything like that. That's also to protect you against infections as a physical barrier. And then you've also got like the acid in your stomach as well, right? And so that is a physical barrier, right? So when you eat something, hopefully the acid in your stomach is going to help kind of kill those bacteria and everything like that, right? And so that is a first line of defense. And so often those physical barriers, your skin, your mucous membranes, the acid in your stomach are not perfect, right? There will be things that get through that. If you get a cut, if you get like a little cold sore in your mouth, stuff like that, or if you just eat enough bacteria, enough or a bad enough pathogen to cause problems, right? So breaking the cycle of transmission is kind of the first step. But if those pathogens, if those foreign invaders, those antigens get through that, we've got these next two steps. So the next thing your immune system is going to try to do is it's going to try to kill the infectious agent. Okay? And so that response is mainly going to come from your phagocytes, so your neutrophils. Remember, those are good for bacterial infections, for eating up those bacteria. And then your monocytes or your macrophages in the tissues. And remember, those are also important for phagocytosis of random junk and foreign invaders and stuff like that. Okay? And so that's what they're going to do when you get a cut or something invades your physical barriers. Those cells are going to be called to the scene, and they're going to try their very best, their very darndest to eat everything up and to get rid of it. Okay? And they do a good job, but again, they're not perfect, right? So if it's a bad enough cut, anything like that, it's going to need the next line of defense, and that's going to be your B and your T cells. Okay, so those are a type of lymphocyte. So your lymphocytes are kind of the next set of defense. And so they're going to be called into the scene. And remember what they do is they are specific to a certain pathogen, right? And so they can't fight against everything like your neutrophils and your macrophages can. That's why they're kind of higher up in the line of defense. That's why they happen first, because they respond to anything and everything. Your lymphocytes will only respond to very specific things that they have been made to fight against. And so that's why they take longer. But once they're there, they do a much better job of it, okay? Because they're designed for that foreign invader, for that antigen, for that pathogen. And then remember, and we'll see in a second, that these lymphocytes, these T and B cells, can form memory T and B cells. And so what that means is the next time you're invaded with that exact same pathogen or antigen, your lymphocytes will remember it. They'll say, hey, I've seen this dude before, I am ready to go. And they'll actually fight it off faster the second time because they've seen it before, they know exactly what they're doing, and they're more alert, I guess you could say. Okay, so it forms acquired resistance, meaning the next time you acquire that disease, you are more resistant to that infection. Okay, and they do that through those memory cells. So, what causes an immune response, right? What is going to trigger this big old response? Get the lymphocytes out there, get the neutrophils and the macrophages going. It's basically anything your body does not recognize as self, right? So normally in a healthy patient, if nothing's gone wrong, right, your macrophages aren't going to start eating up your normal cells, right? And that's because it knows that those cells are a part of you. It recognizes them. They have special receptors that tell them, hey, we're the same, buddy. We're working with the same person. Don't attack me, right? So anything that doesn't have those special receptors, your body will recognize as foreign. It'll say, oh my gosh, you don't have those receptors. You might have some really weird, crazy ones that I'm triggered to be active against. So anything like your bacteria, your viruses, parasites, those kinds of microorganisms, tumor cells. Remember, because tumor cells actually have a mutation. There's something wrong with them, and so they fail to oftentimes express those special receptors that tell your body, hey, I'm a normal, healthy cell. So if they don't have those receptors, then often your immune response will say, hey, something's going on with these cells. It will attack them and create an immune response. Okay, another thing is allergens. So, hey, fever, poison ivy, right? Pollen, that's what this little girl's freaking out about, is some pollen. So what allergies are is basically your body is sensitized to some kind of allergen, and it actually creates more of an exaggerated immune response than it really needs. Okay, so you breathe in pollen and your body says, hey, that's not a part of me, I don't like it, it tries to attack it. And if you keep getting exposed to that pollen over and over and over again, your body will create a really strong immune response. And the next time you see pollen, it'll be like, oh my gosh, here's that pollen again. We freaking hate that pollen. So we're going to attack it, and we're going to attack it hard. And so your body kind of overreacts. That's what allergies are, basically. Autoimmune disorders. So that is when your body does start to attack cells that belong to you. And this is obviously, a type of disease, a type of disorder, right? So this isn't normal. This is when your body doesn't recognize certain cells that belong to you as a part of you. And so it actually creates an immune response to it because it thinks that it's a foreign invader. So that's in some types of arthritis. Your body will actually start attacking those joints. In type 1 diabetes, your body will actually start attacking certain receptors and stuff like that, certain cells. And then it's also in graft rejections, right? So if you get a graft, right, that's another part of a different human. And so if it's not matched well enough, if your immune system is strong enough, it will see that this is not a part of you and it will start to attack that, and your body will reject that graft. Okay, so that's what causes an immune response. It's basically anything that your body, those immune cells, see, and they say, hey, no, this is not a part of us. This is not supposed to be here. We got to get rid of this thing because it's going to cause us harm. Okay, so now we're going to talk about the two main categories of immunity. So there's acquired and there's inherent immunity. So acquired immunity is specific to whatever's invading, and it requires previous exposure to whatever's invading. And that makes sense, right? It's acquired. So we need to have seen it before in order to acquire this type of immunity. And remember, we said that's mainly our B and T lymphocytes, so our lymphocytes, right? And what happens is antibodies are made to a specific antigen, right? So antibodies are made from those plasma cells, B cells, and then T cells are also made to a specific antigen, okay? So they're made for a specific antigen, and they can only respond to that antigen. And so it takes a while to respond the first time, so that's what this is showing. It takes a while to respond the first time they're exposed to that antigen, but the second time you are exposed to that antigen, the response is really hyped up and it is much faster, it's much more effective. And that's how our acquired immunity works, okay? So it's not that great at responding the first time, but chances are if you're exposed to it once, you're going to be exposed to it again. And that's what this acquired immunity is kind of preying on, is the fact that we're going to see it again, and the second time we see it, we're going to come back stronger and better than before. Okay? So this is just showing you, when you inject somebody with an antigen, this is the antibody concentration, right, being made by those B lymphocytes plasma cells. And so it's not very strong the first time, and then the second time, that response is much higher, much greater. That's what we call an acquired immune response. Okay, and then we have inherent immunity. Inherent is just like it sounds, right? So you already have it, it's already present, you don't have to do anything to get it, it's inherently there, okay? And the thing about inherent immunity is that it is generic, right? You don't have to be exposed to the pathogen before. It doesn't really care what the pathogen is. It's going to respond to it because it's foreign, because it's not supposed to be there. And so that's going to be your phagocytic cells mainly, okay? So your neutrophils, your macrophages that come from your monocytes, as well as your physical barriers, right? Your skin is going to keep out what it keeps out. It doesn't care what it is, right? It doesn't care what kind of bacteria, what kind of virus it is. It's going to keep it out, right? Same thing goes with your phagocytes. They're going to eat it up no matter what, okay? And then it protects against and kills many different types of antigens because, again, they don't really care what it is. And it does not require previous exposure, right? It's inherently there every time, all the time. Okay, so now we're going to learn about the different kinds of lymphatic organs that we're going to focus on. So these are the organs that really help your immune system with its function, right? And we just covered its function, so to fight against foreign invaders. So these lymphatic organs are really going to help that happen. And we categorize, because we love to categorize things, we categorize these organs into two big different categories, and that's going to be a primary lymphatic organ or a secondary lymphatic organ. Okay, so our primary lymphatic organs do not need direct contact with the blood. Okay, what I mean by that is it is antigen independent, meaning those organs do not need to come in contact with an antigen or a foreign invader or whatever in order to perform their function, okay? So they are antigen independent. That's a primary lymphatic organ. They do not need to come in contact with antigens in order to contribute their part, okay? So what they do is they produce those necessary immune cells. So they produce the B lymphocytes and the T lymphocytes, okay? They have no afferent lymphatics. That's just a difference that you guys need to know. We'll cover it when we go over secondary. And its function is to produce, right? To make those B cells and those T cells. And so your two primary lymphatic organs that we're going to talk about are going to be your thymus and your bone marrow, okay? And so the main difference between primary and secondary is they don't need to come in contact with the blood, and they actually don't want to come in contact with the blood, and by that I mean those antigens, okay? Because they don't need antigens to be able to make those cells, right? And actually they don't want antigens in order to be able to make those cells, okay? Your secondary lymphatic organs need to come in direct contact with blood or lymph, okay, meaning they need those antigens to be there in order for them to perform their function. And that's because their function is to utilize those immune cells that were made by the primary lymphatic organs, and they utilize those immune cells to bring them in contact with those antigens. Okay, so they take those immune cells, they take those antigens, they mush them together, and they say, here, fight it out, guys. This is where you're going to fight it out, and see who's going to win. Okay, so that's what those secondary lymphatics do. They need those antigens in order to perform their function, in order to bring them in contact with those immune cells. They do have afferent lymphatics or a high blood supply, and the way that makes sense, right, is because we need those antigens that are in the blood or in the lymph. We need them to come into the organ in order to come in contact with those immune cells, right? So we need things coming in. Afferent lymphatics means coming at the organ, right? Or a high blood supply. We need a high blood supply so we have lots of antigen coming in to come in contact with those immune cells. We don't need that in these primary lymphatic organs because we're just producing things. So things are just going out, not in. Okay? And so its main function for the secondary lymphatic organs is to filter, right? To bring the blood, to bring the lymph in contact with those immune cells, filter that blood and that lymph out, so that hopefully we can fight off some of those antigens. Okay, and some examples of those secondary lymphatic organs are going to be your lymph nodes and your spleen. Okay, and so I will tell you guys, one of the things that makes this unit hard is this difference right here. Okay, is this difference between primary lymphatic organs and secondary lymphatic organs. You need to understand that they both work for the immune system, but they have kind of different main functions, right? One is to produce those cells. The secondary lymphatic organs is to use those cells that were produced. Okay, and then another thing that makes this unit hard is we have all of these lymphatic organs, right? They're all going to have a high concentration of lymphocytes in them because they're either making those cells or they're utilizing those cells. And so if you zoom in really closely on that organ, it's going to look really similar to all the other lymphatic organs, right? Because you're just going to see a bunch of lymphocytes. So they look very similar if you zoom in very close. It's hard to differentiate between all the different types of lymphoid organs. And so one of the main things that I need you guys to really get a handle on this week is how to identify what organ you're in. Because if you can identify what organ you're in, you can also tell me a lot about the structures that are known to be in that specific organ. You can tell me a lot about the function of that organ and what's going on. But you can't do that if you don't know what organ you're in, right? Then you're just guessing. So that's my main thing for you guys to focus on, is focus on the difference between a primary and a secondary lymphatic organ, and then focus on the difference between identifying different types of organs. Okay, how you can identify the difference. So this is a diagram of a thymus. This is a diagram of a lymph node. We're going to go over each organ in detail and the differences in just a second though. Okay. So the first kind of lymphatic organ we're going to go over is the bone marrow. And remember, this is a primary lymphatic organ, right? And so when I say it's a primary lymphatic organ, that means it is antigen independent, right? We do not want antigens going into the bone marrow because we're still making these immune cells, right? So that's like... So my analogy for primary versus secondary is you wouldn't want to, this is going to sound weird, but if I'm training police officers, right? That's the same as training these lymphocytes. If I'm training police officers, before I ever set them outside to fight off crime, I want them to know what is good and what is bad, right? Same with my cells. When I make my lymphocytes, I want them to know what is me and what is not me, right? And so if I want them to know what is good and what is bad, what I'm going to do is whenever they're in training, it would not be helpful for me to surround those training police officers with criminals, right? when they're supposed to be learning what's good, when they're supposed to be learning the right thing to do. I do not want to surround them with a bunch of crime because they're not ready to face that yet. They don't know what's good or what's bad yet. Okay, I just want to teach them what's good first. And that's how it works with these lymphocytes, is while they're being formed, I just want to teach them what they can recognize as self. Okay, so if I put an antigen in there, it's going to say, oh, this antigen must be good. This antigen must be self, because it doesn't know it yet, right? So then you get a police officer that's recognizing all these crimes as good things, or a lymphocyte that's recognizing all these antigens as self. So hopefully that makes sense. So that's why we do not want antigens in our primary lymphatic organs, because they're still learning what is self, right? They're still trying to figure out what makes up this person and what this person's cells look like. So if I put antigens in there, they're not going to know the difference between a bad guy and a good guy. Okay, so that's kind of how I think of it as far as that goes. Hopefully that helps you guys. But yeah, so in the bone marrow, it's a primary lymphatic organ. We do not want antigens in there because these cells do not know what is self yet, right? They're still learning, so we don't want to put any antigens, any bad things in there. And what the bone marrow is primarily responsible for is producing B lymphocytes. Okay, so bone marrow, B lymphocytes, bone marrow, B lymphocytes. So hopefully that makes sense for you guys. And then remember, when we learned really briefly about the bone marrow, it makes your other types of white blood cells. So your eosinophils, your neutrophils, your monocytes, everything like that. But as far as lymphocytes goes, it matures and produces your B lymphocytes. Okay. The next one is going to be your thymus. So your thymus produces your T lymphocytes, okay? And if it's producing T lymphocytes, it means it's a primary lymphatic organ, right? Primary lymphatic organ, because it's producing things. It's forming these T lymphocytes, and hopefully you recognize thymus T. I think I already said that, but yeah. So it's a primary lymphatic organ. It has no afferent lymphatics, right? Because we don't need all of these lymphatics going into it, right? We don't need all of these antigens going into it because we're producing these lymphocytes, right? They're coming out. It's antigen independent, right? Because these T cells are still forming, these T lymphocytes are still forming. We don't want to show them antigens yet because they're not yet mature. They're still trying to find out what is self, okay? And then how do you identify a thymus? So this is kind of the important stuff when we get to all these different organs, because they do look very similar. So the way that I identify a thymus is that it is lobulated. Okay, lobulated. So you see how there's all these different kind of divisions between the lobes of the thymus, right? So all of these are different lobes of the thymus, and they're separated. So that's very characteristic of the thymus. And so what it looks like is that there's multiple medullas and multiple cortexes, okay? So it looks like, this is one cortex. So the cortex is the outer part. The medulla is this lighter inner part. So it looks like here's a medulla, here's a cortex, right? Here's a medulla, here's a cortex. Medulla, cortex. Right? So it looks like there's multiple medullas and multiple cortexes. What I will tell you is that there's only one medulla and one cortex, and they do all connect to each other. It just looks like they're separated because it's lobulated, if that makes sense. Okay? It's just because of the way that it's cut. Really, this medulla is interweaving between all these lobes to connect to each other, and same as the cortex. But it looks like there's multiple medullas and cortexes, and that's just because it is lobulated. And that's very characteristic of the thymus. So if you see this kind of lobulated appearance, that's the thymus. Okay? And then when we look closer at the thymus, we'll see that it has continuous capillaries. And remember, that makes sense because when we learn about continuous capillaries, we said that continuous capillaries are used when you want as little contact between the blood and the tissue as possible. And remember, we already said in a primary lymphatic organ, we don't want very much contact between the tissue and the blood because we don't want antigens to get in here and mess up these guys' training, right? The training of these little T lymphocytes. So we have continuous capillaries. And what helps form those continuous capillaries are these special cells called epithelial reticular cells. Okay, we'll see that in a second, but those epithelial reticular cells are what are going to help produce the blood thymus barrier. And then another thing that's very characteristic of the thymus are these things called Hassel's corpuscles. Okay, and we'll see that in a second. So here we can see, so we're a little more zoomed in, and a hint that I will always give you guys is in, I believe, every question there's going to be a zoom out that will show you where we're taking this image from. So if you look at the zoom out and you see this lobulated organ, right, with it looks like multiple medullas and multiple cortexes, it's the thymus, okay? But if we zoom in here a little bit, you can see, so this is going to be that outer cortex region of one lobe. This lighter staining portion is going to be that inner medulla portion of one lobe. And if we see here, so you see these kind of pink blobs, these very light pink, thick blobs, those are those epithelial reticular cells, okay? And then right here is a Hassel's corpuscle. And we'll look at it a little more zoomed in. There you can see there's a good zoom in of a Hassel's corpuscle. So here you can see really well, more zoomed in, and you can see there's a bunch of thymocytes in here. Thymocytes, that basically means cell of the thymus. Basically what they are is they're those T cells forming, right? They're trying to form, and then there's some other cells interspersed throughout there, but the main thing we're going to focus on are these epithelial reticular cells. So you can see it's this really thick epithelial cell. It's this special epithelial cell, and again, remember that helps to create that blood thymus barrier. Okay, so here's another example of an epithelial reticular cell. Here's another example. That's another example. That's just the best looking one. And then here you can see these are those Hassel's corpuscles. Okay, so they're kind of like these little bundles. That's kind of what corpuscle means. They're these little bundles of stuff, and we actually don't really know what they do yet. Okay, we think it's to help these thymocytes mature, these lymphocytes mature, but we're not really sure. So just know how to identify them, not really what they do. Okay, so the next organ is going to be a lymph node. So remember, we already covered the bone marrow and the thymus. Those are our two primary lymphatic organs. So now we're going to cover the secondary lymphatic organs. And so first we're going to cover the lymph node. And so what the lymph node mainly does is it filters lymph. Okay, so it filters lymph. So when we learned about all those lymph vessels last week, and how they're carrying lymph, what they're doing is they're carrying them from one lymph node to the next. Okay, so that's what that does, and it is a secondary lymphatic organ, right? And remember, what that means is it is antigen dependent, right? It needs those antigens to come in contact with this organ in order for this organ to fight those antigens, right? To do its job. Okay, and so because it does rely on lymphatics for its supply of those antigens, it does have afferent lymphatics. Okay, so afferent means going towards, right? So afferent is going at, efferent means going away from or exiting. Okay, and so it does have lymphatics going towards and away from it, because remember, we're filtering lymph, right? So we're bringing it in and letting it go out. So you will see that there are afferent lymphatics going towards the lymph node. So how can you tell it's a lymph node? How are we going to identify it? And the way I identify it is it has one big cortex and one big medulla. Okay, so here you can see in the middle, this is one big medulla. The medulla is more towards the middle. And the medulla has a bunch of blood vessels, right? A bunch of blood vessels and everything all throughout here. The cortex is kind of the more functional part on the outside. So all of this is cortex. Okay, one big cortex. All right. And this will vary through species. Some species do have lobulated lymph nodes, but I don't want you guys to worry too much about that. Okay. And then it does have follicles with germinal centers. Okay, and so this is kind of where it starts to get confusing, because structures like this, I can see how if you'd look at this, you'd be like, okay, well, there's one little medulla and there's one cortex. So there's multiple, looks like multiple medullas and cortexes. So it's the thymus, right? that is falls. This whole big thing is the medulla. This whole big thing is the cortex, okay? What these are follicles within the cortex, okay? So these are follicles within the cortex. They're much smaller. They're much more bundled off. They are going to always be this kind of circular shape. So these are follicles, and what they have are germinal centers. So this lighter staining part on the inside of the follicle, that's the germinal center of the follicle, okay? And that's where we have high concentrations of those lymphocytes and antigens, where they're kind of doing their little battle, okay? And then it also has what we'll call parafollicular areas with high endothelial venules. We'll see that in a second. And it does have afferent lymphatic ducts. Remember, we said that earlier. So if you look on the outside of here, sometimes you'll be able to see some afferent lymphatic ducts. That does kind of tell you you're looking at a lymph node. Okay, so here we've got a little bigger zoom in of a lymph node. And so here you can see all throughout the middle, that is just one medulla, one cortex on the outside. So we've got a lot of blood vessels in the medulla. Hopefully that helps you identify that. And then out here, so this is not a medulla and a cortex, this is a follicle. This is one follicle, this is another follicle, so this big kind of circle looking thing. And these ones do have those germinal centers, so that lighter staining center where they're kind of fighting their fight, okay? And then, so these are the follicles. We also have this thing called a parafollicular area. Parafollicular, right, means like next to the follicle. The follicle. So it's next to the follicle. So if this kind of big circly looking thing is the follicle, the parafollicular area will be right outside the follicle. So this would be our parafollicular area for that follicle. This would be our parafollicular area for that follicle, and so forth. And within that parafollicular area, we'll see in a second, we have these things called high endothelial venules. Okay, high endothelial venules. And then you can also see we have these afferent lymphatics. Okay, so these are the afferent lymphatics. In the lymph node, you have multiple afferents and only one efferent. Okay, so there's multiple ways for this lymph to get into the lymph node. There's only one exit. So the one efferent is going to be very big. The little afferents are going to be kind of lined up all around the side. Okay, so here you can see in an even bigger zoom in, and remember we've got this kind of big zoom out to tell you what kind of organ you're looking at. So hopefully that helps you know that it's the lymph node. If not, you can also look at this, right? And you can say, yes, this looks like a follicle with a germinal center. Okay, follicle with a germinal center, follicle with a germinal center, these kind of circular looking things. And then right outside the follicle is where we have that parafollicular area, right? So this is the parafollicular area of that follicle, and we have these things called a high endothelial venule. Okay. And so what that does, those high endothelial venules, is they make it hard for lymphocytes to exit the lymph node. Okay, so lymphocytes can enter the lymph node, but they can't really exit. The only way they can really exit is by going through the efferent lymphatic duct. Okay, so if you have a bunch of lymphocytes, because remember, lymphocytes are going to be going through the blood, they're going to be made in the thymus, right? They're going to be made in the thymus, they're going to be made in the bone marrow. So if they're going through the blood and they need to get to a lymph node, they can do that. Okay, but if they want to go from that lymph node back into the blood, they cannot because of these high endothelial venules. These high endothelial venules make it to where the only way they can get out is through the efferent lymphatic duct. And if they go out the efferent lymphatic duct, the only thing that they can do is go to the next lymph node, or the next lymph node, or the next lymph node. Right, so now they're in lymphatic circulation and it's really hard for them to get out. Okay, if that makes sense. That's what those high endothelial venules do. And they're these kind of hot pink looking things. High endothelial venules just means they're like fatter, right? They're taller because they create more of a barrier. And then, yeah, there's some more afferent lymphatics. Okay, so more afferent lymphatics. These kinds of thin vessels here with that light pink lymph within them. Okay, so the next one we're going to go over is going to be the spleen. And so remember we said the lymph node filters lymph. It's pretty straightforward. The spleen filters blood. Okay, so they both filter things. They are both secondary lymphatic organs, right? Because remember, the main function of secondary lymphatic organs is to filter things. The spleen just filters blood instead of lymph. Okay, and so it is a secondary lymphatic organ, and remember that means it does need direct contact with blood. It does need those antigens to go through the spleen in order for it to perform its function. Okay, so what happens, or how we're going to identify the spleen, is it has what's called red pulp and white pulp. Okay, and so here you can see this is the diagrammatic view, and these kind of circular looking things are going to be the white pulp, and everything kind of around it is going to be the red pulp. And the reason they're named that is because the white pulp has a higher concentration of white blood cells. So this is where your white blood cells are sitting. They're waiting to come in contact with an antigen so they can fight it off, right? But they need their specific antigen. The red pulp is where all the red blood cells are filtering through. Okay, so that's kind of a big thing that you use to identify the spleen. Another thing is it has central arteries and penicillar arteries. Okay, so these central arteries are going to be closer to or associated with the white pulp. So this would be a central artery, this would be a central artery. I know it's confusing because they're not in the center of the white pulp, but they're associated with the white pulp. So these would all be central arteries. And then the arteries that are off to the side are going to be your penicillary arteries. Okay. And then another thing it has is Billroth strands and littoral cells. And we'll look at that more in a second, but that's kind of what gives structure to the spleen. Okay. And so again, I know this can be confusing because you can look at this and you can very easily get confused between this and other lymphatic organs, right? But another way to look at this is the spleen does not have a medulla or a cortex, okay? So these little white pulp looking things are spattered all throughout instead of, more towards the outside or something like that, like it is in the lymph node, okay? So I know how you could look at this and you could say this is a follicle with a germinal center, right? But it's not. It is white pulp and this is red pulp. So we're in a completely different organ, so that name does not apply. We're in the spleen. Okay, so I know that can get really confusing, you guys. That's why it's really important to know what organ you're in. But yeah, okay, so these are going to be our little white pulp circle looking things where we have a lot of white blood cells. Everything around it, that's going to be our red pulp where we have all of our red blood cells. Okay. And so here you can see in a better zoom in, and so you can see the zoomed out one, right? How there's no big medulla or spleen. There's all these little white pulp things splattered all throughout, right? Same with this one. And if we zoom in, you can see, so this is white pulp right here, right? All of this is red pulp. And you can see how there's this little artery associated with the white pulp. So those are going to be central arteries. Penicillar arteries or penicillary arteries are going to be in the red pulp. And basically the way that works is, because remember we said this is a secondary lymphatic organ, its function is to filter the blood, right? So we need a lot of blood in this spleen in order to be able to filter it, to get out all the antigens, to get out dead red blood cells and junk stuff like that. So what happens is these penicillary arteries, right? Normally when you have an artery, it goes to an arteriole, to a capillary, then it goes back to a venule, a vein, and the heart, right? What happens with these is these arteries just end, okay? They just end like the end of a hose. So they don't go down to the capillary level and then go back to a vein and the heart. What happens is it's like you took an artery and you just cut it. Okay, so it's like an open ending, and what happens is it's like I took a hose and I just plugged it into the spleen. So all it's going to do is it's just going to spew whole blood into this spleen. Okay, so whole blood is just going to get spewed into the spleen. It's going to be forced to bounce around here to be filtered, and that's because we want these red blood cells to be filtered. We want to get rid of the old weary red blood cells. We also want to get rid of any antigens, right? Because there's this white pulp in here with all these white blood cells, and we need them to come in contact with the antigens. So we want the antigens to be floating around in here so that these white blood cells can pick up on that and start eating them up and fighting them off. Okay, so that's how those penicillary arteries work. And so this blood is going to bounce around here and stuff like that. And then it has to exit by draining into those venous sinuses. Okay, so the venous sinuses are these big kind of gaps. These big kind of gaps within the network of the spleen that the blood collects in, pulls in, and then will filter out. Okay, and so the kind of network of the spleen that keeps structure to it are going to be these things called Billroth strands. Okay, and there are special stains where you can actually see the strands very well, but basically what they do is they just give structure to the spleen, because remember we've got just a bunch of blood floating around in here, bouncing around in here, so it's really hard for the blood to, or it's really easy for the blood to all just fall to the bottom, right? And then you just got a pool of blood at the bottom. They're not really bouncing around and interacting with anything, so it's not really doing its job, right? So we want the blood to bounce around. That's why we have all these little strands all throughout here to keep the blood kind of suspended all throughout the organ. Okay, and then we also have these things called littoral cells. Littoral cells, so you can see in a zoom in right here, so this is a zoom in of this, and we'll see a better depiction of it, but basically littoral cells are kind of like a gate. Okay, so they're a gate and they only let mature and young red blood cells through. Okay, so if you are an old red blood cell, you cannot go through the gate, you cannot pass because you need to be eaten up and processed to make way for new red blood cells. Okay. So there we go, and so here, so we can see this is kind of how those littoral cells work in the spleen, right? Because the function of the spleen is to filter blood, and so we have all those white blood cells to come in contact with antigens, but we also need to filter out old red blood cells. And so what happens is we have these littoral cells. So the way I think of it is the two T's in littoral make a gate. Okay, littoral, it's a gate, and so they make a little gate, so they line up like this, so there's only very small gaps in between the littoral cells. And so if you are a new fresh red blood cell ready to work, they are very flexible and they can very easily squeeze in between these holes. Okay, if you are an old decrepit red blood cell and you need to be replaced, you can't function as well, they'll actually get stuck in these holes. Okay, so they'll get stuck. So here you can see this is an old wrinkly red blood cell and it got stuck. So it is stuck there, and what happens is there's a little macrophage waiting in the tissues, or a little neutrophil. The macrophage is waiting in the tissues, and whenever these cells get stuck, it's actually going to come out from hiding and it's going to eat that red blood cell up. Okay, and it's going to recycle its parts so we can make new red blood cells. So that's another function of the spleen, is filtering of blood. We got to get rid of the old red blood cells so we can make more new red blood cells. And so this is just a cross section of the spleen, and it's got that stain for macrophages. So you can see there are a bunch of macrophages all throughout here, and they're just waiting to eat up old red blood cells. Okay. So here you can see more of those reticular fibers I was talking about, right? Because remember we said we're just dumping whole blood in the spleen. We do not want all that blood to just fall to the bottom, right? We want to keep it suspended. So we have all these fibers, these kind of black looking things spattered throughout here, and that's what gives structure to the spleen, so that the blood kind of has to bounce around back and forth through all these little places. Okay. And so this is a special stain. It's a reticulum stain to pick up these reticular fibers, because it's hard to see in other stains, but I promise they're there. They're these black kind of stringy things. And what that does is it has a pinball effect, right? Because if you think of a pinball machine, the whole point is to not let that pinball go to the bottom. You want it to keep bouncing up and stay in the machine, right? So you can keep playing. That's what these kind of strands do, is they make it sure that the blood kind of bounces back and forth and it never really gets to the bottom of the spleen. Okay, so that's done with all of the big lymphatic organs. Now we're just going to talk about other areas where you have concentrations of this lymphoid tissue, this lymphatic tissue. So that basically just means this tissue that has a high concentration of those lymphocytes, right, that are ready to come in contact with an antigen. So places where we're going to have high concentrations of lymphatic tissue are going to be places where we have a high likelihood of coming in contact with an antigen, right? with a germ or something. So they're going to be tissues that are exposed to the outside environment. Okay, so the most common places are in the GI tract, right? Because you're eating just unbeknownst amounts of bacteria, of germs, of viruses, all that kind of gross stuff. You're constantly kind of eating them. So that's a great place for your white blood cells to sit and wait and try and come in contact with an antigen. Okay, so you've got little bits of lymphoid tissue all throughout there. So here we can see little parts of lymphoid tissue, and this is in your tonsil. Okay, so these are the tonsillar crypts. Remember, the tonsil is that kind of dangly thing in the back of your throat, right? So you'd think that'd come in contact with a lot of bacteria, right? So we've got these kind of little nodules or follicles throughout the tonsil, and that's just where you have high concentrations of those white blood cells. Okay, they're getting ready to come in contact with some sort of pathogen. This one right here, this is in the esophagus, right? Because remember, you're still eating things, you're eating germs. So in the esophagus, we also have follicles where we have a high concentration of white blood cells. And they're again hoping that if you are going to eat a germ, they can come in contact with it and know what's coming before it gets to your intestines and stuff like that and actually causes a problem. Okay, so they're just kind of trying to pick up on these germs early so they can start hosting their immune response. Okay, here's another one. So this is in the stomach. In the stomach, and we have these little high concentrations of lymphoid tissue. I believe this is in the large intestine, so we have these big high concentrations of lymphoid tissue. So this kind of follicle-looking thing. Okay, so they're just these kind of follicle-looking things with lots of lymphocytes. Okay, so now we're going to review a little bit to see how much you remembered about, identifying these lymphatic organs and then identifying some things that are found within each lymphatic organ. So we're going to look at this one off to the left, and you're going to try to identify this before I tell you what it is. Okay, so hopefully you're thinking maybe this is a lymph node. Okay, because it is a lymph node. And the way I know this is a lymph node, okay, and it's hard when I don't have that zoom out. Normally you will have that zoom out. But the way I know this is a lymph node is this is a follicle. Okay, this is a follicle with a germinal center, because in lymph nodes the follicles are always going to be right towards the outside. Okay, right towards the outside because they're in the cortex. Right, so these are two follicles with germinal centers. Remember, we've got this parafollicular area, so this area right around the follicles, and that's where we have these high endothelial venules. Those high endothelial venules. Okay, so that's a lymph node. This one right here, try and identify it before I say it. So hopefully you're thinking maybe this is the spleen. Okay, and again, remember, this is why I said this is so confusing, because you can look at this and you can say, hey, this looks a lot like this does, right? But this is not a follicle with a germinal center. This is white pulp, okay? And another way to know it is it's not towards the edge of the organ, right? And it does kind of look different, so it does just kind of take practice. I can look at these really easily and just tell you what organ it's in. It does kind of take practice, just the way that they orient themselves, okay? So we've got this white pulp surrounded by red pulp. We've got a central artery, right, associated with this white pulp. Hopefully you can see how that's different than the follicles in here. So this is the spleen. Some of the structure: white pulp, red pulp, central artery. That's probably another white pulp with a central artery down there. Remember, we've got penicillary arteries in the red pulp. I just can't see a good example of one here. But yeah, that's the spleen. So both of these, hopefully you can tell me, are secondary lymphatic organs. So this one, so we're going to identify these. So take a look at these and try and tell me what you think they are. So hopefully you're thinking these are both the same thing, if that helps you. Hopefully you're thinking this is the thymus, okay? And so this one is a lot easier for me to identify as the thymus. That's because it is lobulated. You see these big old separations going in there? those are the different separations of the lobes of the thymus, right? So hopefully that really helps you to identify this, right? It looks like, so here's a part of the medulla, here's a part of the cortex out here, medulla, cortex out here, right? And that's again because it's lobulated, okay? And then if we zoom in, remember what we said we see in the thymus are going to be these epithelial reticular cells, and remember that helps make the blood-thymus barrier. And then these little bundles called Hassel's corpuscles. Those bundles called Hassel's corpuscles. And then a bunch of thymocytes. Okay, so now we're going to do some practice. So what are the functions of the lymphoid system? Okay, so A, protect against foreign invaders into the body. Yes, that sounds right to me. Remember, that's what we've been talking about this whole time, is protecting against those foreign invaders. Protect the germ-free environment of the body. Yes, remember that's what we've kind of been talking about as well. And then protect against type 2 diabetes. No, your immune system does not protect you against type 2 diabetes. Type 1 diabetes is actually a result of an overreaction of your immune system, of your lymphoid system. But yeah, it definitely doesn't protect you against any type of diabetes, and it's just not related, so it would just be D. Okay, which characteristics of immunity best match the corresponding definitions? Okay, so acquired, when we think of something that's acquired, do we think about the response being unique to each exposure? And not necessarily. Let's go to the next one. If we think about specificity, do we think about something requiring exposure to the antigen? No, not necessarily. So the way that I would think about this is you could maybe kind of think these are right until you read both answer choices and you think, hey, no, acquired kind of sounds like it requires exposure to the antigen, right? That's kind of what matches. Specificity kind of sounds like response is unique to exposure. So these are actually switched, okay? Because remember when we said it's an acquired immune response, that means we have to have had exposure to the antigen before in order to acquire that response, right? Specificity was us talking about how the T and B cells, the T and B lymphocytes, their response is unique to each antigen, okay, to each exposure. Okay, and then memory remembers the previous exposure. That one makes sense. Okay, so it would just be C. Okay, so now we've got some matching, and this is kind of tricky, right? This is what I was telling you guys about. So, which organs of the immune system best match the images? Okay, so A, it's trying to tell me this is the thymus, and you can see in the zoom out right here. So this doesn't really look like the thymus to me, right? It's not like one big lobulated looking thing. Yeah, it doesn't really look the same as what I'd consider a thymus. This is actually the tonsil. Remember, we looked at it really briefly. This is actually the tonsil, and we've just got these tiny little concentrations of lymphoid tissue, but it's not the same as the thymus where we have a medulla and a cortex and all of that, and it's all lobulated in one big kind of jumble. So this is not the thymus. Okay. B, it's saying this is a lymph node. And yes, I agree with that because we've got this follicle that's towards the outside of the organ, right, in the cortex. We've got a follicle in the cortex, and remember that follicle has a germinal center. We've got those high endothelial venules. And then we've got these afferent lymphatic vessels as well. Okay, so that all does seem like stuff that would be in a lymph node, so that one is correct. This one, it's saying, is the spleen. And remember, in the spleen, we have red pulp and white pulp, right? This, to me, doesn't really look like red pulp and white pulp, right? We don't have, like, those Bill-Ross strands or anything. And here, right here, you can even see a zoom in. This is a Hassel's corpuscle. And remember, we said those Hassel's corpuscles are in the thymus, and that's where we've got those epithelial reticular cells, we've got thymocytes, and so this kind of lighter staining portion would be the medulla of that one lobe. This kind of darker portion would be the cortex of that one lobe. So this is the thymus. Okay, so the only one that matches would be B. Okay, here, which lymphoid system characteristic best matches? So my advice would to you guys would be identify the organ first. And this is a really tricky question, but it also is a really good way to test whether you know how to identify these organs, because these are the three that look the most similar. Okay, so this one right here, all of them have this kind of lighter staining, like circle, outer looking thing, right? They've got these two different like textures, right? But you need to know the difference between the organs. So this one right here, the circular looking thing, that could be a follicle or white pulp or a medulla, it's more towards the outside, right? It's in the cortex. So this is a follicle, right? This is a follicle with a germinal center. So this is actually a lymph node right here. So this is all cortex, and this is the medulla of the lymph node. So this is a lymph node. Okay, so when we think about lymph nodes, do they have afferent lymphatics? Yes, they do, because remember we said lymph nodes filter lymph, so we have lymph going in and out of the lymph node, so that is correct. B, so we're going to try to identify this one, and this one, if you look at it, it doesn't really look like it has one big medulla and one big cortex, right? These kind of circular looking things are kind of spattered throughout. They're kind of everywhere. There's not a big separation between medulla and cortex like there was out here where the follicles are all on the cortex. All right, so this is actually the spleen, right? And these are white pulp and red pulp. So this is the spleen. And so yes, what this one's saying right here, red pulp and white pulp, yes, that does match for B because this is the spleen. C, it's saying Hassel's corpuscles. So C first, let's try to identify. So we can obviously see right here, right? We've got these big separations. That's defining the different lobes of this organ. So this is the only lobulated organ we're talking about. So this is the thymus. And remember, these are all the medulla, the cortex of the different lobes of the thymus, okay? And so remember when we did learn the thymus, it does have those Hassel's corpuscles within it, okay? So it is all of the above, but this one is tricky. Hopefully this helps you kind of juxtapose the different organs and see how, yes, they do look very similar, but also different, and I do need you guys to know the difference, okay? So it's tricky. As always, this doesn't cover absolutely everything you're going to need to know this week. I did just try to hit the big topics, but hopefully this was helpful for you guys. Hopefully this is going to help you differentiate between all the different organs and what's going on with them and what the immune system really does. But yeah, good luck on your quiz.